Again, to tell people, my guest is Dr. Darryl Felder, professor of biology here at UL Lafayette. He is also head of the Crustacean Research Laboratory here at the university, and also is part and editor of a pretty amazing series called Gulf of Mexico, Origin, Waters, and Biota. He was directly involved with the first volume, Biodiversity, and that is becoming quite a Bible for a lot of people right now, trying to understand what the baseline is. He just recently got a National Science Foundation, a RAPID grant with his colleague Suzanne Fredericq, and they will be going out to study some of the effects of the oil spill on, in particular, your area of research, crustaceans, and Dr. Fredericq's algae. And by algae, I think some of us have this sense of just, I don't know, little tiny things in ponds, but she's talking about seaweeds. I mean, big seaweeds, red algae. And
they're big and they're little, they're green and they're red, and they're brown and they're yellow, and they are incredibly abundant when you get out there. They're coating the surface. They're like little meadows.
And a lot of creatures live in those little meadows, as she was explaining. Probably a lot of your crustaceans live out there.
Some of my crustaceans will occur only on a certain species of her algae. So we have a little symbiosis going here.
Right. So sketch out for us once you get out on the boat in November, and then you'll probably go twice. I think you're scheduled to do 2 trips out there. What are you going to be doing when you get out there?
We have a total of 10 days, so we can split that in 2 cruises of 5 days each, or do it in one 10-day batch. It's going to depend in part on how we can orchestrate vessel use, but the area that we're going to be targeting this time, while we previously surveyed the entire Gulf of Mexico, that's way more than we can afford to do with our initial funding. We may do a much more extensive survey when we come back and make another proposal to the NSF. But for right now, we're going to be targeting northern Gulf of Mexico banks. There are a number of these, obviously, directly south of Lafayette. Some people know about areas out there, but you get out to the outer continental shelf, get to the margins just before where it's dropping off, and kind of get east of the area that people know as the flower gardens. And there's a lot of pinnacles and banks that fishermen know, and some are in diving depth, but the ones we're working on are below that. And then as you go east, you begin to hit some more off the mouth of Mississippi. Many of these have been targeted for years by fishermen. They know that they're good places to catch grouper and snapper, even though they're fairly deep. Sackett Bank is one right off the mouth of the Mississippi River just to the west. And then you come around the Mississippi River, and there's a number of them off Alabama. And then eventually you get toward Florida, and there's an area known as the Florida Middle Grounds. Now, some of the middle grounds is very shallow. It's coralline, and we stay off of that. It's a preserve. We obviously stay out of the reserve, so we're on deeper banks than that. So when we get out there, we have something called an hourglass box dredge that was developed many years ago by the Florida Department of Natural Resources to do initially a survey off West Florida. And that was kind of an early database on these kinds of banks. So we deploy this as steel boxes dropped over, dead slow. The engine is barely going in and out, and it collects like several buckets full of rubble from the top of these pinnacles. And then that's brought on board, that's dumped on a sorting table, and we turn the students and graduate students, the doctoral students and postdocs loose on it in addition to ourselves, as long as our backs can take it. But it goes on day and night. And you process this, you bring it in to the lab on board the vessel where things are actually sorted and preserved. Some are photographed immediately because color is often very important in the identification. But then they're adequately preserved, so when we get them back to the lab, we can gene sequence them, because we've got to get them back to the lab. We're going to extract DNA. We're going to run what's called a PCR to replicate that DNA. We're going to sequence it, and we're going to be able to tell you what this organism was by comparing it to large banks of computer data for previous sequences. So that's kind of the scenario. And so in the end, we know exactly what was there. And if we get an odd sequence, we may have discovered something that was not there before, or we may find a subtle difference from one bank to the other that says, you know what, this is the same species, but this population is different. And then you say, well, is it different physiologically? Is it something that's at risk? So the questions can get real complex once you get into the DNA.
And obviously it takes a lot of patience.
Takes time.
I'm sure some people must wonder, why do you have to do all the DNA sequencing? Can't you just tell what things are? But as you mentioned, there are something like 1,000 species in the crustaceans just that you're interested in. And how many different species, when in your book, Biodiversity, and the whole series on the Gulf, I think you told us a number one time of the different species.
Right. When we put this together, there were, of officially named species that are included in the book, are 15,300 and some odd. I don't remember the exact. But, everyone that contributed said, well, these are the ones I know that are good names for. I know of another 100 of my group. In my own case, there's 1,010, I think, is what we're officially in the book, and I know for a fact there's another 50 to 60 more already.
Not named.
Not named. But more than this, when you get to groups like the microbes, I was talking to you before we went on the air about Dr. Shestasardov. He contributed to this. When you get into those groups, they don't even have a naming system the way the traditional systematists do anymore. They've given up a long time ago. And there are thousands and thousands and thousands of what you could call species, and they're simply identified by a genetic code now. And so that's one way of addressing a group that's more complex than we ever imagined.
Well, we'll have to have him come on and talk about some of the changes that may be going on in the Gulf in his area. But again, we are speaking about, in particular, crustaceans and algae right now that you and Dr. Fredericq will be studying. You all received this RAPID grant from the National Science Foundation. And I guess it's called RAPID because it makes them able to respond very quickly to situations like this and get money out to scientists like yourself.
Exactly. It's got an absolute limit on it of $200,000. But it does not go through the normal process of review. In other words, normally when you turn in an NSF grant, it may be out there for 3 to 6 months being reviewed by scholars all over the world. They do more of an in-house review. I think they send it out by email to several experts. But I really don't think you stand a chance of getting this if you haven't been previously funded by them. So they already have faith in your ability to produce.
Right.
And that's what they want to invest in.
Yeah, it says a lot about your past record and what they think of your scientific methods. So this, again, this will take place over 2 voyages out into the Gulf, and then you'll bring that back. But as we mentioned earlier, you are hoping to write further grants, and I mean, obviously this is going to take a lot more than just 2 trips out into the Gulf. And as much as we would like to know the answers now, and like to know what the effect is on the crabs and the seaweed and everything, it's just not going to be known for a long time.
No, NSF even knows that. When they fund these, let's get you started quickly, because we can tell this is something we're going to want to know. We were the first. I don't know if there have been any others that were funded in the Division of Environmental Biology for NSF, and I think they were looking for, okay, what can we do? What is something we do well in our division that we can fund? And we just happen to fit. And but as soon as they saw our proposal, they got back to us and said, okay, we're going to fund this, but you guys better come back with something longer term. We know we can only fund you for a year under this thing. So they've all but invited us to come back with a longer-term proposal.
Right.
And it's not that, and you and I were talking about this before, too. It's not that Suzanne nor I want to define ourselves as oil spill specialists. And, that's not how we got into this business. But it's certainly impacting the groups of organisms that we've been studying. And one of the reasons we studied them was because they were potentially vulnerable. So I think we're doing it.
You've been preparing yourselves for this moment.
We have,
also.
And we kind of owe back to NSF the commitment to try to make the data as relevant as possible in this setting.
Well, of course, there's always the curse about, the curse is may you live in interesting times.
They are interesting.
It would be so much nicer to be studying just healthy creatures and just enjoying the biodiversity of the Gulf. But your talents are being called to study something that's not so pleasant. And
so long as we're studying recovery, once it stops and we're studying recovery and we can see rates of recovery, my optimism will be there. there.
Right. Then you'll know the baseline at least,
Right.
or you'll be able to study what the baseline is. Is there a place where people can go to find out a bit more about your work? I mean, at the book we mentioned Biodiversity, I think, is how many pages? Like 800?
Right. No, that's almost 14,000 page
14,000
book, so
pages.
that's not light reading, and
Right.
it's pretty much a reference book. Maybe the introductory chapters and some things would be of interest to folks.
Well, can you suggest some place for more or less lay people?
I would. There is a website called the Gulf Base website that's maintained by the Hart Institute for Gulf of Mexico Studies at Texas A&M Corpus Christi. Just type in Gulf Base and search that. There's all kind of references there that they'll point you toward. It's a nice synthesis for what's going on in the Gulf of Mexico. It's got some local things in it, but some more national, international as well. One of the nice things about that institution is it bridges beautifully to Cuba and Mexico as well, and so it's truly international. It believes in all partners in the Gulf of Mexico playing a role, which is another thing that made me enthusiastic about working through them for this. And then each of us, Suzanne and I have our own websites, and if you just search our names, you can track us down. I don't, I'm supposed to have my website committed to memory. I don't know. I push a button on my computer.
Well, just search UL Lafayette Crustacean Research,
Right.
and then Suzanne, of course, is the Seaweed Lab, and it's very nice. All of you do have nice photographs of what you're doing, and really gives a sense of all of the students, as you mentioned earlier, that you have working with you, graduate students, doctoral students, and it is a huge department. Remind us again how many professors you all have. And
well, we have, I think, 28 now is the total number, and most of those are very research active, and a number of them are oriented toward specifically marine issues. So we have a pretty good-sized group that's targeting marine issues in the Gulf of Mexico now.
All right. If you have just tuned in, Dr. Darryl Felder has been my guest. He is a professor of biology at UL Lafayette, head of the Crustacean Research Laboratory. Dr. Suzanne Fredericq and Dr. Felder have received a National Science Foundation grant. It's a RAPID grant to go out into the Gulf and to study the impact of the oil spill, particularly on their area of study, crustaceans and algae. And perhaps we'll be able to have the two of you back after you go out on your first voyage in November.
Be happy to do so.
Thank you so much.
Thank you.